This video will be explaining about the three main processes related to DNA, which are DNA replication, DNA transcription, and DNA translation. And in each process, the main enzymes/protein that plays the major role will be emphasized. DNA replication. The basic mechanisms of DNA replication are similar across organisms. DNA replication is semi-conservative, meaning that each strand in the DNA double helix acts as a template for the synthesis of a new complementary strand. From one DNA double helix molecules to two DNA double helix molecules, with each newly formed DNA double helix containing one new and one old strand. The DNA replication process will start when DNA helicase, which is the unzipping enzyme, catalyzes the unwinding of the double-stranded DNA. When unzipping, it breaks through the hydrogen bonds that hold the DNA bases together, which then forms the replication fork. This will produce two strands, the leading strand and the lagging strand. The leading strand runs from the 3' to 5' direction, while the lagging strand runs from the 5' to 3' direction. Each strand is bound by a single-stranded binding proteins that binds to single-stranded proteins and stabilizes them so that they don't come back together. Each DNA strand will then act as a template of replication. The enzyme topoisomerase will prevent the DNAs from supercoiling. The enzyme primase will catalyze the addition of RNA primers. These primers will act as a markers to help guide the DNA polymerase III enzyme and initiate the replication process. DNA polymerase III then binds to the primers and catalyzes the addition of complementary DNA bases to the 3' end of RNA primer. The DNA polymerase always need a template. They can only add nucleotides to the 3' end of a DNA strand. They can't start making a DNA chain from scratch, but require a pre-existing chain or short stretch of nucleotides called a primer. One new strand which runs 5' to 3' towards the replication fork. This strand is made continuously because the DNA polymerase is moving in the same direction as the replication fork. This continuously synthesized strand is called the leading strand. The other new strand, which runs 5' to 3' away from the fork, is trickier. This strand is made in fragments because as the replication fork moves forward, the DNA polymerase, which is moving away from the replication fork, must come off and reattach on the newly exposed DNA. This tricky strand, which is made in fragments, is called the lagging strand. The small fragments are called Okazaki fragments, named for the Japanese scientist who discovered them. The DNA polymerase I, which is also known as the exonuclease, will remove the RNA primers and replace by DNA to continue the process. Then, the gaps between the DNA fragments are sealed by DNA ligase and joins the Okazaki fragments together. DNA transcription. Transcription is the process in living cells in which the genetic information of DNA is transferred to a molecule of messenger RNA. This process occurs in the nucleus and involves three stages, which is the initiation, the elongation, and the termination. The enzymes involved are the RNA polymerase, which is to unwind the DNA double helix and catalyzes the adding of RNA nucleotides to the base pair of the DNA template. The RNase sync enzyme to remove introns and join exons to create an mRNA molecule with continuous coding sequence only. The first stage, which is the initiation, starts at the regions of DNA called promoters. The promoters will determine which strand of DNA is used as a template. From 3' to 5'. Next, RNA polymerase binds to the promoter and it catalyzes the unwinding of DNA double helix and separates the two strands of DNA apart. Only one strand of DNA acts as a template. Then, RNA polymerase catalyzes the adding RNA nucleotides to the base pair of the 3' to 5' end of the DNA template. The second stage is the elongation stage. RNA polymerase moves downstream from 3' to 5' of template. The RNA polymerase catalyzes the adding of RNA nucleotide, forming a growing RNA strand, which is complementary to the DNA template. The RNA transcript mRNA is formed from 5' to 3' direction. Behind the point of RNA synthesis, the double helix reforms and the RNA molecule peels away. The transcription process ends during the termination stage when RNA polymerase reaches a terminator sequence of the DNA. Once reached, RNA polymerase stops adding RNA nucleotides to the RNA strand and detaches from the DNA template. The new mRNA molecule is released and then it leaves the nucleus and attaches itself to the ribosome complex to begin translation. DNA translation. DNA translation is the process of translating the genetic code carried by mRNA in the form of codon into a sequence of amino acid in a polypeptide chain. Polypeptide is then further modified into a specific protein. This process requires the messenger RNA, the ribosomal RNA, or the transfer RNA. The enzymes involved are the amino acid tRNA synthetase to catalyze amino acid activation, peptidyl transferase to catalyze the formation of peptide bond. It involves three stages, which is the initiation, the elongation, and the termination. Before mRNA is translated, amino acid activation, which is the process in cytoplasm where amino acid binds to tRNA forming amino acid tRNA occurs. Each amino acid will join to tRNA by the enzyme amino acid tRNA synthetase. Then, a specific tRNA with a particular sequence of anticodon will bind to a specific amino acid, which code is complementary to anticodon of the tRNA. The translation will start with the initiation stage. During initiation, a small ribosomal subunit will bind with mRNA. Then, initiate a tRNA with anticodon UAC that carries amino acid methionine binds with start codon AUG on mRNA. Initiation factors will then bring in the large ribosomal subunit, causing the initiated tRNA to occupy the P site. This will make the A site empty, available for the next amino acid 3RNA. The next stage is elongation, which involves the addition of other amino acids to the initial amino acids that result in the growing of polypeptide chain. It involves three steps. The first step is the codon recognition. The anticodon of an incoming tRNA with its amino acid will pair with the mRNA codon in the A site of ribosome. The second step is peptide bond formation. The growing polypeptide chain will leave the tRNA in P site. It becomes attached by a peptide bond to amino acid on the tRNA at the A site. Peptide bond is then formed between carboxyl terminal at a growing peptide from tRNA in the P site and amino terminal of amino acid of tRNA in the A site. Peptide bonds between the first and second amino acids are formed through a peptidyl transferase activity. The last step is translocation. Ribosome will move one codon ahead, causing tRNA in the P site to be translocated to the E site where it is released. while tRNA in the A site translocated to the B site. The next amino acid tRNA with anticodon complementary to the next codon in mRNA will come into the A site. This process is repeated until a complete polypeptide chain is formed. The third and the last stage of DNA translation is the termination. It occurs when one of the three stop codons UAA, UAG, UGA reaches this A site, or A site reaches one of the three stop codons, which is UAA, UAG, UGA. The release factor will bind to the stop codon and hydrolyzes the bond between the polypeptide chain and its tRNA in the P site by adding water instead of amino acid to the polypeptide chain. This frees the polypeptide and translation complex deassembles. In conclusion, there are a total of 11 enzymes which play important roles in the processes of DNA. 7 in DNA replication, which are the DNA helicase, single-stranded binding proteins, topoisomerase, DNA primase, DNA polymerase III, DNA polymerase I, and DNA ligase. 2 in DNA transcription, which are the RNA polymerase and RNA splicing enzyme, and another 2 in DNA translation, which are the amino acyl tRNA synthetase and peptidyl transferase.